we have, you all know that there are two kinds of nucleic acid, deoxyribonucleic acid and ribonucleic acids. Both the nucleic acids are made up of monomers which are called nucleotides. RNAs vary, the length of RNA, all three kinds of RNA vary from 100 to many thousands of nucleotides, whereas DNA molecules are much, much longer. When we come to the definition of gene, what is gene? It is a unit of DNA that contains information for the synthesis of a polypeptide chain. Up till now, we have studied gene as a unit of heredity, but since it is molecular biology, so here the purpose of gene, what we are going to study, it is heredity of course, but when you are studying, it is hereditary unit. But when we are studying molecular biology, the main function of gene is to provide information so as to synthesize a single polypeptide chain. The unit of DNA which has the information for the synthesis of a polypeptide chain, that segment of DNA is called a gene. And this synthesis of DNA to protein, it takes place through another molecule which is ribonucleic acids. All the three kinds of ribonucleic acids are involved in formation of polypeptide chain. So the protein coding genes, they work through messenger RNA molecules of the cells. DNA itself does not send the information directly to uh, the protein synthesis machinery. It goes through messenger RNA molecules of the cells. This is just the now what is central dogma? Many a times short note comes on central dogma. You must know central dogma is formation of mRNA from DNA, that is transcription, and formation of polypeptide chain from mRNA, that is translation. This is known as central dogma. That is DNA to mRNA, mRNA to polypeptide, transcription and translation. First transcription occurs and second step is translation. This is known as central dogma. The three processes that will lead to production of protein is replication of DNA, then transcription of messenger RNA from DNA, and then translation of messenger RNA to polypeptide chain of amino acids. These three processes together constitute the production of proteins. During DNA replication, as I said earlier, DNTPs, they are polymerized so as to form two identical copies of each chromosomal DNA molecule. We all know DNA exhibits three characteristic features. The first amongst the three characteristic features is semi-conservative model, which itself shows that semi-conservative means that when DNA is going to replicate, one is going to be the parental strand and another will be the new strand. Every DNA double helix, one has to be the parental strand, the other has to be the daughter strand. So that is known as semi-conservative molecule. Now, so as that each daughter cell receives identical copies. Now, what is transcription? In transcription, RNA polymerase, they come and together and form precursor messenger RNA molecule, which is commonly called pre-mRNA or primary mRNA molecule. By the polymerization of ribonucleoside triphosphate monomers called as RNTPs. Okay, hope it is clear. Now, what is RNA processing? RNA processing is actually the removal of non-useful mRNA part. It is removed and the final product, the mature mRNA, is transported to the cytoplasm. So during translation, the language of protein is the amino acid. In transcription, the language was bases. DNA is also made up of bases and mRNA is also made up of bases, nucleotides. Whereas in translation, the language is of proteins. That is why it is called as translation, because the language is different now. mRNA has the language of nucleotides, whereas protein has the language of amino acid. Therefore, it is called as translation. Now, the entire concept of protein synthesis occurs on ribosomes. We all know the function of ribosome is for synthesis of protein. Ribosomes are made up of rRNAs and multiple proteins. After transport to cytoplasm, these ribosomal subunit, the smaller and the larger subunit, they associate with messenger RNA. And then they further, with the help of tRNAs and rRNAs, synthesize. That is a detailed chapter that will be done later. Now, in DNA replication, one thing is to be kept in mind, that DNA are anti-parallel. Means 3' end will face 5' end. On the opposite end, 5' end will face 3' end. That is the phenomena known as anti-parallel. Anti-parallel. Had it been parallel, it would have been 3' end, it would have been 3' end. But anti-parallel, 3', then the opposite 5'. Now, what happens in DNA replication? This helix, it unwinds, and in the process, one DNA strand is continuous and the other DNA strand is discontinuous. Continuous DNA strand is called as leading strand and discontinuous DNA strand is called as lagging strand. The direction of synthesis is always going to be from 3' to 5'. This is 3 dash to 5 dash, so this is going to be the direction. This is 3 dash to 5 dash, so this is going to be the direction. This is another diagram which will make it all the more clear. The enzyme used for DNA replication is DNA polymerase. Simultaneously, on both the strands, DNA replication occurs. As I told you, one is going to be the leading strand, other is going to be the lagging strand. In lagging strand, DNA segments are synthesized in pieces and then these pieces are joined together. That is, it is discontinuous replication. Whereas in leading, it is a continuous replication. These subunits, they are not to be joined further. It is in a form of a continuous strand. We can see this chapter later on in detail. This is just to remind you of the entire process. These are the enzymes which are used in replication as well as transcription. Helicase is the enzyme which opens up the DNA strand by breaking the hydrogen bond between them. So the DNA strands have to open up in replication as well as in transcription. Ligase, as we all know, ligase is for joining. So it is used when these fragments, the discontinuous DNA strand fragments known as Okazaki fragments, they are united with the help of enzyme ligase. Primase is the word derived from primer. So this enzyme helps in the synthesis of RNA primer. Endonuclease are the enzymes which attack in between the DNA strand, not in the corners. Otherwise, it would have been known as exonuclease. But it cuts the DNA strands in the middle. So it is known as endonuclease. Single strand binding proteins, commonly known as SSBP, it binds to the single strand of DNA so that it does not rewind again into its secondary structure. To prevent it from rewinding, these single strand SSBP proteins, they bind to the single strand of DNA. Now in this slide, we will see the exact process of transcription, which is the main topic. DNA is in the form of double helix. As I said earlier, it has anti-parallel strands. One of these strands is going to act as a template. Other strand is going to act as the coding. So this is the coding strand and this is the template strand. mRNA will form at template strand rather than the coding strand. Why does it happen so? Because the code which is present in the coding strand is required for protein synthesis. And how will this code go to the mRNA molecule? Because the anticodes of this coding strand is present on the template and the anticode of template will be present on the mRNA. So the sequence of nucleotide mRNA will be sequence equal to the sequence of same as the sequence of the coding strand. Our purpose is to have this sequence which is present in the coding strand to reach the mRNA molecules, and it will only reach the mRNA molecule through the template strand. So template strand is going to have complementary base pairing as compared to the coding strand, and mRNA is going to have complementary base pairing as to the template strand. So this base pairs on the mRNA will be same as that on the coding strand. So same information which is present in the coding strand, that is transferred into mRNA, except that thymine is replaced by uracil, which is a characteristic feature of RNAs. Now, The coding strand, this is the coding strand. As you can see, the coding strand runs from 5 dash to 3 dash direction, whereas the template strand, it runs from, this is the template strand, and as you can see, it runs from 3 dash to 5 dash direction. Either it is replication, or it is transcription, or it is translation. The thumb rule is that the whichever strand is forming whichever strand, it is always going to be anti-parallel. If it is replication, the daughter strand is going to be anti-parallel to the parent strand. If it is transcription, mRNA is going to be anti-parallel to the template strand. If it is again translation, then tRNA is going to be anti-parallel to mRNA. Wherever it is, the other strand has to be anti-parallel to the previous strand. So in this case, the template runs from 3 dash to 5 dash. So mRNA formed will be running from 5 dash to 3 dash end. This is from 5 dash to 3 dash end. And a short segment where the DNA and RNA, they stay together, that is known as the hybrid region. And RNTPs, they come, they and polymerize to form the mRNA strand. Now what is this? This is unwinding and rewinding of DNA. What is unwinding? Because the direction of polymerization is going to be from, in case of template strand, from 3 dash to 5 dash. So this is going to be the movement of the polymerase enzyme. So this is the direction of unwinding, and opposite side is going to be the direction of rewinding. These are the areas of supercoiling, which we have already studied in DNA replication about supercoiling. So in the direction of polymerase movement, the DNA helix, it unwinds, and the part which has already participated in RNA mRNA formation, it rewinds. This is the nature of DNA. So transcription has 3 steps: initiation, elongation, and termination. Initiation is just the beginning of transcription. Elongation is when the RNA strand, it increases in length by joining of new RNTPs. And termination is when this polymerase enzyme reaches a particular sequence, that is the termination of mRNA. Now we are going to see these all 3 steps one by one in detail. Now, as I said earlier, we are talking about RNA polymerase. As I said earlier, during transcription, one of the DNA strand acts as a template and the other strand acts as the coding strand. mRNA is formed on the template strand. Just a minute. mRNA is formed on the template strand, not on the coding strand. This you have to remember. The direction of template strand is going to be 3 dash to 5 dash. Therefore, the direction of mRNA formed is going to be from 5 dash to 3 dash. 5 dash end of mRNA is going to be the mature end, and the newly synthesized end is going to be the 3 dash end. That means the RNTPs, they are going to arrange themselves at the 3 dash end. There they will polymerize at 3 dash end rather than the 5 dash end. 5 dash end of mRNA is the mature end of mRNA. The bases in template DNA strand, they base pair with the complementary incoming RNTPs. That means if it is going to be C in DNA, cytosine in DNA, then in RNTP it is going to be guanosine. If it is going to be adenine in DNA, Then in mRNA, it is going to be uracil because thymine is not present in mRNA. It is replaced by uracil. And all this arrangement, the enzyme required for this is known as RNA polymerase. All the polymerization reaction, all the joining of RNTPs will take place with the help of enzyme known as RNA polymerase. Now this polymerization, it ends at the 3' end of RNA. This we will see in the next diagram. See here we see this is the DNA template. These are the bases arranged. As I said, it is going to be complementary base pairing. Template is always to be in 3' to 5' direction, whereas mRNA will be formed in 5' to 3' end. This is the 5' end of mRNA and this is the 3' end of mRNA. Always, as I said, polymerization is going to occur at 3' end. So this is the mature mRNA section and this is the nascent mRNA section. Means the first nucleotide which must have arrived at this polymerization is this and the last is going to be this. So DNA direction is 3' to 5'. Now what I mean to say is that the next RNTP which is going to come, it is always going to attach itself at the 3OH of the previous base. See, this is attached, this is incoming RNTP and it is going to attach 3-OH end. This attached to 3-OH end. This attached to 3-OH end. So the new RNTP is going to attach at the 3-OH end. This is coming, it is going to attach at 3-OH end of the previous base. Now let us study in detail the 3 steps. The first step is the binding of polymerase enzyme to the DNA. Now first thing you make clear that the polymerase enzyme, it does not attach to single strand of DNA. It will attach to the double helix of DNA, right? RNA polymerase always binds to the double strand of DNA double helix, not to the single strand of helix. And the place at which RNA polymerase binds, it is known as the promoter. So the RNA polymerase binding at the promoter along with the DNA double helix is known as the closed complex. It is known as the closed complex. On the DNA double helix, one is the start point, other is the stop point. On the template strand, so this is the start site and this is the stop site on the DNA template. That means which is running from 3' to 5' direction. Now what does this polymerase do? This polymerase unwinds this DNA double helix, which is called the melting of DNA duplex. Where does it melt? At the start point. Why would it melt at the start point? Because RNTPs, they are going to begin the transcription at this start point. So the DNA double helix has to unwind at this start site on the template strand. Now this, when this DNA melts, this is known as transcription bubble. In replication, it is known as replication bubble. In transcription, it is known as transcription bubble. And now the polymerase, this transcription bubble and the start point, all this together is known as the open complex, right? Now polymerase, it catalyzes the formation of phosphodiester linkage between two RNTPs. What is the function of polymerase? Is to catalyze the linkage between the two initial RNTPs. What is the linkage? What is the kind of linkage? It is the phosphodiester linkage. As in DNA, similarly in RNA, it is going to be the phosphodiester linkage. Next step is elongation. Now, as I told earlier, the part of the DNA helix which has RNA still attached to it, that is known as DNA RNA hybrid region. The part which moves out and the part which remains attached. The part which remains attached of the RNA with the DNA, that is known as DNA RNA hybrid region. Polymerase always moves on the DNA strand, on the template strand from 3' to 5' direction. But yes, of course, it remains attached to both the strands of DNA, not with single strand of DNA. Termination will happen when the polymerase is going to reach the stop site. As it reaches the stop site, it releases itself from the double helix and the complete RNA strand is also released. So the RNA which is formed is from the direction of 5' to 3' end. I hope this is clear. So what is initiation of transcription? When RNA polymerase forms a transcription bubble. Polymerization of RNTPs will start at the start site, which is located in the promoter region. Where will polymerase attach itself? To the promoter region. What does promoter region has? Start site. What will happen at start site? RNTPs will come and polymerize. Once the DNA region that has been transcribed, the separated strand, they will again rewind and form a double helix. And the RNA which is formed will be released from the DNA template strand. Only that part of the RNA will remain attached where new nucleotides, where new RNTPs are coming and polymerizing. And that will be occurring at the 3' end. So 5' end will be released and 3' end will always remain in the hybrid mode with DNA. So 5' end of RNA will exist. Termination will occur when the polymerase will reach a particular sequence which is known as the termination sequence, and that is known as the stop site. So during transcription initiation, RNA polymerase recognizes double strand, that is the promoter region of the double strand. Other factors are also required along with transcription, along with RNA polymerase. These are known as general transcription factors. They help RNA polymerase to locate the promoter. How will the RNA polymerase identify the promoter region? With the help of general transcription factors. After binding to the promoter, RNA polymerase melts the DNA strand so as to make it accessible for RNTPs to come and polymerize. Now, what is the size of the transcription bubble? About 14 base pairs of DNA, they melt, and that is the region of the transcription bubble. What is the length? 14 base pair. And what is the length of the hybrid? 8 base pairs. That is the hybrid region, and the bubble region, transcription bubble region, will have 14 base pairs of DNA around the transcription site. As I said, transcription bubble is a 14 base pair. Ribonucleoside is added at the 3' end of the mRNA molecule. 5' end will be released and polymerization is going to occur at 3' end of the growing RNA strand. Termination is the final stage in RNA synthesis. Complete RNA molecule, or which is called as primary transcript, is released from the RNA polymerase and polymerase dissociates from the DNA. So what will happen? The primary transcript is released from the RNA polymerase and RNA polymerase in turn is released from the template DNA. All the three things, they move away from each other. What? DNA template, RNA, pre-mRNA and the polymerase. All three of them, they segregate after the primary transcript is polymerized. When will this step happen? Only when the polymerase reaches the termination sequence. The released RNA polymerase will again go to the same gene or other gene and start synthesizing next mRNA. It will recycle itself, it will become a part of the cycle again and again.